Hey everyone, how's everyone doing? I'm just waiting for a couple more minutes to see if other people turn up. We'll aim to start at 5 past. Right, let's get started. So, we've got one person in the wait. So today's topic is on pathogens and infectious diseases. So it's in a variety of specs. I think it's in, and I know it's definitely in the OCR, EdXL, and WJEC specs. I'm not sure if it's in that much detail in the AQA spec. If anyone's in the AQA spec, let me know. But I know this is definitely a focus point in the other specifications. So, pathogen is any organism that can cause diseases. So there are many different types of pathogens, such as viruses, bacteria, protocysts, and fungi. Can anyone give me a type of virus, a type of bacteria, a type of protocyst, and a type of fungi? MRSA. So what's MRSA? Is that a bacteria, a protocyst, a virus, or a fungi? E. coli, yep, that's a bacteria. Yep, so flu. Flu is a range of viruses. Salmonella is a bacteria. Malaria is a protocyst. Athlete's foot is obviously a fungus. Measles is a virus. MRSA is a bacteria, and MRSA is a bacteria. Tobacco mosaic virus, yeah. So the first plant-based one. We've seen all the other ones so far have been mainly human or animal ones. This is particularly a plant-based one, tobacco mosaic. Same with black spot fungus. Influenza is the virus that causes flu. Yeah, so flu itself is a disease, but the virus is the influenza virus. COVID as well. Obviously, that's a virus, coronavirus. An infectious disease is a medical condition that's caused by a pathogen. So usually infectious implies that they're communicable or transmissible, meaning they can be passed on between host organisms. Sorry, I'm just trying to get to slides. Yeah. So, for example, we've got all these different types of viruses. So HIV, AIDS is human-based virus, influenza is an animal-based virus, and tobacco mosaic virus is plants. Then you got bacterial meningitis for humans, cholera for humans, TB for cattle and animals, including humans, and then ring rot for plants. Malaria, animals, late blight for plants, specifically tomatoes and potatoes. Athlete's foot for humans, ringworm for animals, black sigatoka for plants, specifically bananas. And so here we've got a range of examples of conditions and diseases that are caused by the different types of pathogens. And I would recommend you just getting that down into your notes quickly. I'll give you a second to do that, because examples usually do tend to be quite important in illustrating your points in exams. So we've got different types of methods of transmission. We've got direct and indirect transmission. Direct is the pathogen are passed on directly between one host to another. Indirect, so that could be from droplet or sexual transmission, like HIV or AIDS. Whereas indirect is when the pathogens are spread via an intermediary source before reaching the final host. So that's specifically where you've got a vector, so or a spore or food and water. So viral vectors or parasitic vectors. So that's malaria, for example, is a good example of this. Spores are fungus, and food and water like cholera or salmonella. So yeah, cholera is an infection by the bacterium Vibrio cholerae, and it infects the small intestines, causing watery diarrhea. Fomites are a specialized type of spore that are transmitted through the air. So I think they're included as part of the spores. Cholera is most commonly spread through eating uncontaminated food and water that is contaminated through feces. So it's the oral fecal route, which basically means that you consume the bacteria, then you excrete it, and then you recycle it, and it recycles back. Is this session useful for AQA students? Does anyone know? Any other AQA students? We can take a quick look. I'll look it through the spec, see if there's a module on diseases, but I think this is mainly for OCR students and EdXL students, because I know this definitely comes up in those exam boards. Yeah, but I can check for you. Let's take a look. Genetic information variants, that's not in there. Energy transfers. Might be in cells. Yeah, so you do actually need to know about T lymphocytes, B lymphocytes, role of antibodies, vaccines, antigen presenting cells, structure of HIV. Yeah, topic 2C. So you do need to know a lot about the cell recognition and the immune system. So yeah, this will all come up. We're just touching on the introduction to pathogens. Most of this lesson is going to be spent talking about the immune system and how we respond. So yeah, you do need to know about that. Malaria is caused by parasites of the Plasmodium group. So Plasmodium vivax is the main one, or Plasmodium falciparum. So Plasmodium malaria, Plasmodium ovale, or Plasmodium vivax, they're parasites. And basically what happens to them is they lay eggs within mosquito, as in they are transmitted within mosquitoes. Mosquitoes inject the eggs into humans, and then basically the eggs grow in humans, they reproduce within the humans, and then the eggs are taken back up, or the fully formed version parasites are taken back up by the mosquito and spread to the next organism. So it's a vector-borne disease. It's spread to humans first through female mosquitoes, which are first infected by the parasite, and then they enter the liver cells in the red blood cells. They then are passed on to another mosquito, which are then passed on to another person. So there is a cycle to this. Yeah. As the transmission of malaria requires mosquitoes, especially a specific genus of mosquitoes, female Anopheles mosquitoes are the most common spreading malaria. It's only common in tropical and subtropical regions. These areas have the climates necessary for mosquitoes to breed. Malaria is considered then to be endemic because you have a constant risk in those regions, but you don't have a very big risk outside of those regions. The problem with this is that with climate change, there's evidence to suggest that malarial mosquitoes are now spreading to parts of southern and middle Europe and going further north, because the areas that they normally lived in are getting too hot, and so they're moving further north. And last summer there was first early reports of there being malarial mosquitoes found in the UK. So it could become slowly endemic to other parts of the world with global warming. Tuberculosis is caused by a species of Mycobacterium. Mycobacterium tuberculosis causes TB in humans. And so what happens, the bacteria embeds itself within the apex or in the top part of the lungs, and it reproduces in there, and it causes damage and inflammation to the top parts of the lungs. It's an airborne disease, and it's transmitted via droplets. So it's like COVID, cough-based transmission. And remember, therefore, because it's spread directly, close contacts of people who have the active disease are at a higher risk of becoming infected themselves. The problem with tuberculosis is that it can remain in quite in a latent stage for a significant portion of time. So it can stay in a non-active state in your lungs and then be activated by another precipitating event, which makes it really difficult to kind of diagnose, because it's only diagnosed when it becomes active. So it becomes a little bit harder to manage. Okay. HIV is caused by a specific type of virus, the human immunodeficiency virus, and it attacks key cells of the immune system, which we'll talk about in a couple of minutes. But it specifically attacks, does anyone know? Maybe someone's got some wider reading on this. CD4 T helper cell, yeah. So it attacks T helper cells. And we'll talk a little bit about what T helper cells are, and it lowers the number of T helper cells, and so indirectly it lowers your immune system to everything else, basically, because it slows your immune system down significantly. So over time, then the infected person is going to develop some form of acquired immunodeficiency. Basically, they're not going to be able to respond to a specific another infection or another disease that comes then that's present, which then exposes the person to other infections and cancers, which may become fatal. So that's the problem, and it's transmitted through bodily fluids. So remember, the fluids must come into contact with the into the bloodstream. Basically, they need to enter the bloodstream, and that's how HIV works. So main transmission routes, we have sexual transmission, which is the most common, or direct injection into the bloodstream via the use of unclean needles by intravenous drug users, vertical transmission between a patient, infected mother and her baby, and breastfeeding. Yeah. So HIV can be transmitted by sexual contact, sharing needles to inject drugs, mother to baby transmission, but it's not transmitted by air or water, or saliva, sweat, and tears. or insects or pets, or sharing toilets, food, or drinks. Yeah. Right. That's kind of an early introduction into the immune system. We're going to talk a little bit about plant cells now, plant diseases. So stem rust or black rust is a fungal infection. It commonly affects wheat and barley. And remember, these are going to have serious consequences because they lower the they lower the harvest and yield, which means that you can't eat food that's rotten, and hence it then will have serious consequences on farming and agricultural communities. Where does a virus come from? Like before it spread, how is it there? Well, it's evolution, right? So you started with you started with bacteria, viruses, animal cells, all the different types of cells, and then those viruses have slowly mutated into their varying forms of viruses that all have different DNA and different mechanisms of action. They've just come from the common progenitor, like every life form has on the planet. But technically viruses are non-living because they don't have but anyway, so if we think about this, what factors are going to make something disease easier to spread? Diseases usually spread rapidly in densely populated or unsanitary areas. So if you think about it, like if we think about what kind of factors have spread diseases such as COVID, we know that diseases have spread by people being in a lot of close proximity. So they tend to spread very fast in places where there are loads of people in very close proximity, where not everyone might be following heavy sanitation. So countries that are able to invest in good healthcare systems and public health education campaigns are probably going to be more successful in preventing diseases. Yeah, because you're more educated on hygienic practices and you're aware of sort of treatment and vaccination programs. Now, someone's entered the waiting room. So, for example, with cholera, one of the best things that we can do is we can provide clean drinking water. We can educate people on hand hygiene and hand washing, provide clean lavatories, and better sanitize your communities. But this is kind of crucial. Like the first bit is provision of clean drinking water is probably the most important part. Yeah. Next week's session is on DNA and DNA replication, probably also going to be on ATP because they're all included in that kind of group. Malaria is best treated by destroying mosquito habitats or preventing breeding of female mosquitoes, or public education on using mosquito repellent and mosquito nets. TB, can you think of the factors that are now gonna, okay, let's give some examples of some factors that are going to affect tuberculosis. We know that it's a droplet-based infection, so can we think of some things? So can we think of some things that will affect it? Face masks, yeah. Face coverings. What else? Hand washing, yeah, proper hand hygiene. Social distancing. I mean, probably not likely that they're going to use social distancing. The big one is ventilation, effective ventilation. I know social distancing is one that we do with COVID, but if we think about the more, remember, any mechanism that you use has got to be acceptable for an extended period of time, right? COVID, we did it because it was an emergency circumstance. It's unlikely that for the next 50 years, we're going to have to socially distance. because that would not be acceptable to the population for such an extended period of time. So any public health measures that you do try and introduce have got to be acceptable for the period of time you're intending to use them in. Cleaning surfaces, for example, that we can always do anyway. Isolation, again, probably not. It's going to be very difficult to put in for an extended period of time. Face masks, again, if we can build that culture, maybe. Hand washing, specifically, ventilation, provision of vaccines usually, or treatment on TB treatments. And then, yeah, educating people who have TB to cover their nose and mouth, and maybe for those people to then avoid contact when they have active disease. So if they're in latent disease, they'll probably not have to do that either. HIV, usually it'll be on safe sex and on needles, on needle education, but everything, as you can see, most of it is about public education. So as you're probably aware, healthcare education has become incredibly important, and is a very vital way of explaining kind of ideas and kind of how you're approaching certain things. Okay, so we're going to run through quite quickly on the primary defenses against pathogens, because we're then going to do introduction to the innate and adaptive immune response. So yeah, so we're gonna quickly run through primary defenses. So primary defenses are to stop the pathogen entering into your bloodstream. So this isn't once the pathogen is already there, how do we deal with it? This is how can we stop it from getting there? So can people think of some examples of good primary defenses that humans have? Yep, hair depends. Hair on your head, probably not, because there's no crevice in your hair, but hair in your ears and nasal hair, yeah, potentially. Cilia in the throat, yeah. Scab or clotting, yeah. Blood wound formation, obviously all of those things. Skin, stomach acid tears, skin flora. Nose hairs, yeah, specifically nose hairs. There's a natural inflammatory response. Does anyone know what inflammation is? We'll talk about it, but so the skin basically forms a physical barrier, stopping pathogens from entering the cells. And this is strengthened by a protein that you might have heard of in your basic biological molecule structure lessons, keratin. The chemical defense is the secretion of antimicrobial fluid onto the surface of the skin. Inflammation is a natural protective process to pathogens. The fundamental features of inflammation are going to be redness, heat, swelling, and pain. So these are the things that are most commonly associated with when an infection comes in. Can anyone think of why this happens? I'm pretty sure some people might be aware of why each of these happen and what function they supply. Okay, so redness because of blood vessel dilation. Yeah, perfect. So it's basically the blood vessels dilate, so you have more fluid in the area. Again, heat is also due to that blood vessel dilation. Remember that's quite important because, yeah, swelling because of tissue fluid. Remember, the whole idea is that all of these processes bring together the white blood cells. And when they bring together those white blood cells, they bring the white blood cells to the right areas, and that causes correct kind of inflammatory response, which then causes the right white blood cells to be there at the right time. Okay, the swelling occurs as damaged cells basically release these molecules that cause vessels to dilate, which brings white blood cells and other key components of the immune system to the affected site quite quickly, so that if any pathogen did manage to get in, it can be fought off quite quickly. And then we also then have quick wound formation, which basically means that no more pathogen can get in. And we don't need to worry about blood clotting, but for some specs, you might need to know about it. So blood clotting is to plug the wound, and basically it's all the white blood, the platelets, basically they're tiny cell fragments, and they come together and they fuse with a protein called fibrin, and fibrin forms this kind of mesh. And then basically cells called fibroblasts basically repair this wound, and they repair the skin, so that skin kind of forms underneath this wound, this the scab formation. And then once the skin is repaired, the scab can just shed off. Please, can you recap the mechanisms for inflammation? Yeah, of course. So going back. It's a natural protective response. The swelling occurs as damaged cells release molecules. So basically, molecules are released by the cells around the skin that's been damaged, and basically these molecules, histamine is one of them, yeah. And this causes the blood vessels in the skin to dilate. Remember, we know that there are blood vessels that are literally on your skin surface, and those dilate. And that basically means that if you remember how tissue fluid is formed, based on that pressure gradient, basically there's more things that are kind of pushed out of those gaps in the capillaries. And so there's more tissue fluid around, and that tissue fluid is high in white blood cells and other key components of the immune system, so that any pathogen can be fought off quite quickly. Vascular permeability is vascular meaning anything to do with blood vessels, permeability meaning the ability for anything to get through. So increased vascular permeability is basically the blood vessels have an increased ability to let things get through it. That's what it means. And remember, this increased tissue fluid then causes all of those symptoms: swelling, heat, and pain, and redness. That's what you get when you've got an inflammatory response. Okay, don't worry about any of this. Mucous membranes. So we have a lot of we have mucus lining the membranes. Goblet cells secrete this mucus, and that traps the pathogens. And then ciliated cells sweep these pathogens back out. And so then these trapped organisms can be removed from the body via these expulsive reflexes, which include coughing, sneezing, and vomiting. They occur quite fast when a certain part of the body is irritated. So sneezing occurs quite fast when the respiratory tract is has got some kind of irritant on it. Again, the gut also has a highly acidic environment, which destroys and denatures many pathogens. But there are also other kind of gut bacteria that are present that prevent the pathogens from dividing and proliferating. Plants also have loads of different chemicals and physical responses. So they've got a waxy cuticle. The waxy cuticle is kind of like the skin. It makes it more difficult for pathogens to gain entry inside the plant. The cuticle also prevents accumulation of water, which prevents transmission, because transmission of pathogens between plants. Cell walls, again, a secondary barrier. It's very difficult to break down the cell wall. And then there are other physical defenses. There is callus. Callose is a chemical that basically comes and sticks in between cell walls and the plasma membrane. So they're between the cell wall and the plasma membrane. They're kind of like, what's that thing that people put around housing? Kind of like cladding, or there's a term for it, but yeah, it's to prevent anything from entering into the cell. Basically, callus is also present at the plasmodesmata, which prevents pathogens from spreading cell to cell. And there are different types of chemical defenses as well. Saponins are chemicals that destroy the fungi cell membrane. Phytoalexins inhibit pathogen growth on the cell surface. Caffeine is really bitter, so it prevents any pathogens. It's also toxic to a lot of animals. So it prevents them from being eaten. Yeah. Okay. Immune system, introduction to. And so this is just an overview of the innate and adaptive immune system, and then what an antigen is. So basically, the immune system works through a system of recognition mechanisms. So basically, it recognizes itself and the pathogens, and then it eliminates the pathogens. So there are two main mechanisms. You've got your innate immune system, which is the first line. It's a non-specific immune system. It's general mechanism, and it involves lymphocytes, which break down pathogens via a mechanism called phagocytosis. The adaptive immune system is your second line of defense, but it's a long-term, highly specific and targeted response against a particular pathogen. It involves antibodies, B cells, and T cells. But because the innate immune system is non-specific, it also means it takes less time to activate. So it's a much faster, your initial response has always got to be an innate response and then an adaptive response. So the primary immune response is pathogen enters the blood. Body then detects foreign antigens. Body detects foreign antigens on the surface of pathogens. Neutrophils in the blood break down these pathogens by phagocytosis. And if it's not neutrophils, what other type of cell is present that could do the same function? Macrophages, yeah, perfect. Macrophages are in the lymph nodes, also carry out phagocytosis. Phagocytes are non-specific and they can engulf any foreign organism. And so once they're done, the phagocytes activate. Yeah, they're different phagocytes. They're not the same. Phagocytes activate three types of T cells, and those T helper cells are activated by B cells are activated by T helper cells, and they produce antibodies. So you've got all these different types of responses. Yeah, and B cells then produce further plasma cells, which break down further which make further antibodies. And so that's the adaptive immune response. Okay, so an antigen is a specialized type of protein that is a signaling protein. It's specific to particular cells and pathogens. So all cells have antigens on them. The body cell will have self-antigens. Pathogen will have non-self antigens. The majority of antigens do not exist in normal cells. However, in certain systems, you can get basically the body's immune system attacking its own cell, and that can lead to autoimmune conditions. We'll talk about MHC later. You don't really need to worry about too much about that. And autoimmune disorders, again, they can be acute or chronic, and they can also be long-term, regional, or systemic. So they can be in a specific place They can be all the way around the body, or they can be long-term treatable diseases. The immune system of the host will, if you're having a transplant, the immune system of your host can immediately reject an organ, donor organ. So remember, the donor organ has got to closely match the antigen profile of any recipients that are taking up this this organ. MHC genes vary between individuals except identical twins, hence why you can't take an organ from one person and just automatically put it in another person. You would have to give them some sort of immunosuppressant to basically nullify their immune system. Foreign cells, even if they are human cells, can be recognized as non-self. And that's what the biggest problem is. So we'll talk about the innate immune response first. Have we got any questions so far? We give everyone a minute or two to just collect, to recap what they've done and finish writing out their notes. Any questions? All right, carrying on. So the innate immune response. So can anyone try and list all of the different cells of the innate immune response first? Let's see how many you can get. So we already know macrophages and neutrophils. The innate immune response, remember, is the non-specific immune response that involves phagocytosis. We just talked about it. So it was the very fast one. It's the first form of secondary defense, and it's very fast. It's non-specific to the non-specific to the pathogen. It will attack all pathogens exactly the same. I think we just talked about it, or did you miss that bit? Basophils, eosinophils, yeah, they're good examples. Okay, so cells of the innate immune system can be found in many areas. So we've got mast cells. Mast cells' functionality is to release histamine. Histamine increases inflammation. Don't worry about NK cells. They're not that important right now. Don't really worry about monocytes too much. Neutrophils and macrophages are phagocytes, so they do phagocytosis. And basophils and eosinophils attack specific types of pathogen. So eosinophils are really activated in parasitic infections, for example. Remember, the cells of the innate immune response mostly travel the body via the lymphatic system. So does anyone remember what the lymphatic system is? What is lymph? Yeah, lymph is drained tissue fluid. Sorry, I was slightly lost because I haven't heard some of these terms before. I'm doing it here. Are you in AS? No, you're in A2. That's surprising. Most of these terms, so this is for an OCR spec reference. This is OCR module 4. It's module 4.3, I think, or 4.2. Is this like the primary response for OCR? Oh, okay. Yeah, sorry, there's a lot of time differences on this. Yeah, so this is not the primary defense. This is still the secondary response, but this is, yeah, the non-specific immune response is another term for it. Innate immune response is another term for it. If you guys just list all the terms that you haven't heard before, I'll try and define all of them first for you, and then we can move on. Because there are a lot of terms that are interchangeable. For example, you can use innate immune response. It's the same as the non-specific or the phagocytic immune response as well that other people use. Basophils and eosinophils. Basophils and eosinophils are just specialized types of immune cell. They're basically just generic white blood cells. You don't really need to worry too much about their function. All that they do is that they target specific types of pathogens and defeat them. Neutrophils you've never heard of? Okay, so macrophages, as you know, are your traditional antigen presenting cell. So macrophages, basically they do phagocytosis and then they present the antigens from the pathogen on their cell surface membrane, whereas neutrophils don't do that, but they can still perform phagocytosis. So they're just a different type of cell to perform phagocytosis. You've heard macrophages, AQA. I think because people are in different points with where they are learning at school, maybe, because I know these are all kind of normal types of cells to know. A2. Oh, that's surprising. I'm not sure. So basically, neutrophils are actually the most common type of white blood cell, and they're found in both the blood and in the tissue fluid. They're made in the bone marrow, and they're short acting, and they basically start the immediate response. So they start this non-specific immune response. So the non-specific immune response is started by neutrophils, whereas macrophages are less common, but they are significantly bigger than neutrophils. They develop in the lymph nodes and are made in bone marrow, and basically they're involved as a relay between this non-specific immune response and your specific immune response. So your non-specific immune response is controlled by neutrophils, macrophages, and other white blood cells. Whereas lymph nodes, whereas macrophages are very specifically the connection between that and then the adaptive or specific immune response. Yeah, I know. I was OCR and my school went through all of these terms before because, yeah, they were key terms, but other schools might do things differently. So phagocytosis is the process by which cells engulf pathogens and internalize them, and basically it's the way cells break down pathogens by taking them within to their own cells and then breaking them down. So let's think of this process. So this is an important process for you guys to be aware of. Pathogen is first detected by a phagocyte. Don't worry about the specific names of these terms. I think that might overcomplicate it a little bit. But basically, the pathogen, it will be detected by the phagocyte. So the pathogen is basically the antigens that are present on the pathogen cell surface is detected by specific receptors on the phagocyte itself. The phagocyte then is activated. So the phagocyte basically detects this and says, "You're not meant to be here. Okay, now I'm now activated to kind of destroy you." The phagocyte then takes up this pathogen. So what it does is it basically expands its cell surface membrane to come around and engulf this pathogen. So it takes the pathogen up and it puts the pathogen in a very specific vesicle called a phagosome. A phagosome is a specialized type of vesicle. Yeah. Lysosomes break down. Lysosomes, as you are aware of from your cell structure unit, are specialized vesicles with enzymes in them. And these enzymes will bind to the phagosome, releasing the enzymes, and then the enzymes can perform specific reactions to break this pathogen down. So remember, the enzymes in the lysosome binds to the phagosome and it breaks the pathogen down. Now, there are two ways to do this. The next bit is basically twofold, depending on the type of pathogen you're at. So a neutrophil will release the pathogen or the digested products of the pathogen back into the cell surface membrane, back out of the cell surface membrane and into the blood, where to be excreted normally by the liver or the kidney. Whereas a macrophage will do two things. Some of them they will release, but other bits, like small bits of antigen, they will continue to keep. And these antigen bits will then be taken up and will bind to a specialized receptor. And when it binds to this receptor, that causes the macrophage to become an antigen presenting cell. Okay, so don't worry about, basically all you need to remember for the process is the pathogen is detected, pathogen becomes activated, the phagocytes engulf the pathogen, basically they expand their cytoplasm to go around it, and the pathogen is stored into a vesicle called a phagosome. The phagosome binds with a lysosome, and that makes something called a phagolysosome. It's in the name, phagosome plus lysosome, phagolysosome. And this releases hydrolytic enzymes which break down the pathogen. The phagocyte then absorbs these digestive products. So some products are either basically released, but others are loaded onto specialized receptors. You don't need to know their names. To receptors, and used in the antigen presentation process to activate T helper cells. Okay, does that make sense? Let me know if there's anything that you want me to go over. It's looking familiar now? Cool. Does exocytosis need ATP? It's good question. I'll find out. I don't think so. But actually, no, it probably will. What happens during antigen presentation? I think it does. Someone's already asked this question. Yeah, exocytosis and endocytosis, both requiring the expenditure of energy or ATP. So yeah, fusing with the plasma membrane, I think, isn't the bit that needs ATP. What do you mean by are some contents released? And another question, what happens during antigen presentation? Well, so the antigen is presented. I'm not going to tell you now. I think just for the sake of the class, I'm not going to do the adaptive immune response today. I'm going to try and find another session on it after Christmas, ideally when I plan my next semester's content. And I will give you guys a session on that. But for now, because I've noticed that some of it is still a bit confusing right now. So I'm thinking it might be best just to do a little bit more of a question answer stuff right now. And I will go on to the adaptive immune response in after Christmas. But what happens after antigen presentation is T cells become activated. T cells become activated. That activates B cells. B cells release antibodies, and they form the main memory cells. What it would are some contents really? What do you mean by some contents? I don't know what that kind of. The recording of last week's lesson is on YouTube right now, so you can, if you go right, like let's do this. I'll show you. Go onto YouTube. You go onto a site called Study Mind, a channel called Study Mind, which I'm not subscribed to, but they've got. If you look on this, then you go here. They've got the recording of all of last week's session, so it should be there. When lysosomes, yes. So some of the contents, all the waste products get released by exocytosis, and the antigens become presented. Yeah, that's perfect. That's exactly what happens. Again, there is a little bit more to it, but you don't need to worry about that more because I don't want to complicate the whole class. But yeah, if I probably would go into a little bit more detail if I was, going for a more individual approach. But yeah, I think I don't want to confuse other people. Let me know if you guys have got any questions about the whole topic as a general, even if it is about the adaptive immune response and you've already done it in school or something and you have questions on it. Let me know because I can help you with that now, but I don't want to teach it without kind of being in without not confusing people. If there are no questions, I'm sorry we didn't get through a lot more today. I would have loved to go through other kind of topics, but adaptive immune response is quite complicated in itself. There's a lot of it, humoral. There's a lot of key terms as well, and I didn't want to overdo it. So I'll find a time next semester. I'm now also on the WhatsApp group. So if you guys are on the WhatsApp group, you can send me any questions that you guys have after you've done a lesson or something, or during the week. Just put a message in, I'll try and answer it. So that's more for open Q&A. You guys aren't allowed to type on the WhatsApp group. Oh, that's a bit weird. Am I allowed to type on the WhatsApp group? Yeah. Oh no. Oh yeah. Oh, thought you guys. Okay. Yeah. So if you've got any questions, then bring your questions to the session. How do you join the WhatsApp group? I can send you a link, maybe, if I can. Yeah, I can. Give me a sec. I'll get it to you now. I'm assuming this is the button. Oh no, this one is already full. Yeah, it's kind of weird because there are over 200, there are nearly 300 participants, but no one has turned up yet. So It's a bit like, okay. Should be on in the next couple of days. Yeah, I'm hoping he gets put up in the next couple of days. Again, that will be other medic mind will put that up. Yeah, that was a good lesson on periodicity. Join us on Wednesdays as well for chemistry stuff. There will be more going on. What are we doing in chemistry next time? I can tell you that right now. Give me a sec. Let me look for it. Give me 1 second. Next week in chemistry, it's energetics and equilibria. So we'll be doing Le Chatelier's principle. We'll be doing Maxwell-Boltzmann distributions. We'll be doing all of that stuff. I will send you the, yeah, I'll send you the WhatsApp link here. Give me a sec. I'm just trying to load it. One sec. That's the link to the WhatsApp group, by the way. Hopefully that helps. Yeah. Cool. Otherwise, I will, if anyone else doesn't have any questions, I'm gonna close the meeting. Thanks. Bye.